Hello there! This video presentation will inform you about the principle, general outline, advantage, and disadvantage of enzyme-linked immunosorbent assay, which uses a solid-phase enzyme immunoassay to detect the presence of a ligand in a liquid sample, using antibodies directed against the protein to be measured. Enjoy the presentation!
What is immunoassay technology? Immunoassays are a group of analytical techniques which are used in clinical, biopharmaceutical, and environmental fields. They are analytical methods based on specific immunoreaction between an antibody AB and an antigen AG for the determination of the amount of either reactant in the solution. Immunoassay techniques are suited for the analysis of compounds at low concentration since their detection limits are usually within nanogram to picogram range. Immunoassay capitalizes on the specificity of the antibody-antigen binding found naturally in the immune system. The assay can be used to identify the presence of pathogens in a clinical sample, or it can be used to measure the amount of a target biomolecule. Terminologies used. Antigen is a substance that can be bound by an antibody, capable of stimulating an immune response, specifically activating lymphocytes, which are the body's infection-fighting white blood cells. In general, there are two main divisions of antigens recognized: foreign antigens, or heteroantigens, and autoantigens, or self-antigens. Antibody, also called immunoglobulin, a protective protein produced by the immune system in response to the presence of a foreign substance, called an antigen. Antibodies recognize and latch onto antigens in order to remove them from the body. A wide range of substances are regarded by the body as antigens, including disease-causing organisms and toxic materials such as insect venom. Here are different types of immunoassay techniques. The first one is radioassay. Here, a radioisotope is attached to an antigen of interest and bound with its complementary antibody. The radioactivity of the sample is measured. The amount of radioactive signal is inversely related to the amount of target antigen. The radioactivity concern pushed for alternative methods. The second one is counting immunoassay. Polystyrene beads are coated with many antibodies complementary to the target antigen. During incubation, the beads bind to multiple antigens and group together into a large mass. Some beads remain unbound. The entire solution is passed through a cell counter and only the unbound beads are counted. The number of unbound beads is inversely proportional to the amount of antigen. The third one is fluoroimmunoassay. The antibodies are labeled with fluorescent probes. After incubation with antigen, the antibody-antigen complexes are isolated and the fluorescent intensity is measured. The fourth one is called chemiluminescence. Luminescence is the release of light due to an electron being kicked up to a higher energy state and emitting a photon as it relaxes down. This is the same principle as fluorescence. The difference lies in the mechanism of kicking the electron up to a higher energy in the first place. In fluorescence, this is achieved with certain frequencies of light. In chemiluminescence, this is achieved by a chemical reaction. These reactions require an emitter and a co-reactant. The fifth one is enzyme-linked immunoabsorbent assay, also called ELISA. In an ELISA, the antibody is linked to an enzyme. After incubation with the antigen, the unbound antibody is washed away. The bound antibody enzyme attached to the target antigen is observed by adding a substrate to the solution. The enzyme catalyzes a chemical reaction of the substrate to produce a quantifiable color change. The amount of antibodies are measured in the solution.
General outline for enzyme-linked immunoabsorbent assay or ELISA. Number 1, immunoassay critical success factors such as sensitivity, throughput, dynamic range, reagent cost, etc. The acquired reagents are antibodies, analyte, standards, sample matrices, control samples, labels, and substrates. Number 3, instrument testings are calibration, Performance testing, linearity. Number 4, assay proof of concept experiments. Establish preliminary assay parameters, reagent suitability, and matrix compatibility. Signal-to-noise data analysis models consult immunoassay expert. Number 5, assay optimization. Select factors to be optimized, determine matrix effects, construct precision profiles, consult a statistician. Number 6, assay validation. Robustness, day-to-day effects, scale-up, automation, reproducibility, operator effects, percent recovery analysis. And lastly, method documentation, SOP, and implementation. Enzyme immunoassay method and principles. ELISA is a plate-based assay technique designed for detecting and quantifying peptides, proteins, antibodies, and hormones. In an ELISA, an antigen must be immobilized to a solid surface and then complexed with an antibody that is linked to an enzyme. Detection is accomplished by assessing the conjugated enzyme activity via incubation with a substrate to produce a measurable product. The most crucial element of the detection strategy is a highly specific antibody-antigen interaction. Enzyme-linked immunosorbent assays operate on principles very similar to other immunoassay technologies. ELISAs rely on specific antibodies to bind the target antigen and a detection system to indicate the presence and quantity of antigen binding. In order to maximize the sensitivity and precision of the assay, the plate must be carefully coated with high-affinity antibodies.
So after knowing the basic methods and principles of enzyme-linked immunosorbent assay or ELISA, let's now know the different types of ELISA techniques employed in different applications. So the first one is the sandwich ELISA, wherein the surface is first coated with a solid phase antibody or AB. The test sample containing the antigen or AG is being measured, is then added and allowed to react with the bound AB. Any unbound AG is washed away, and a known amount of enzyme labeled AB is then allowed to react with the bound AG. Any excess unbound enzyme link AB is washed away after the reaction. The substrate is then added and the reaction between the substrate and the enzyme produces a color change. The amount of visual color change is a direct measurement of the specific enzyme conjugated bound AB, and consequently, the AG present in the specimen is tested. So, the sandwich ELISA technique is so called because the AG or the antigen is being assayed is held between the two different antibodies. So, we have here the diagram of sandwich type ELISA. The yellow colored Y-shaped stop there is the antibodies, and the green one is the antigen being sandwiched in between the antibodies. And as a substrate is added, a reaction occurs and a color change is observed, which is the yellow one named as the colored product. This color change is the measurement of enzyme-bound antibody present in the sample. The next type of ELISA is known as the competitive or direct ELISA. The test specimen containing the antigen is to be determined and mixed with a precise amount of enzyme-labeled antigen, and both compete equally for a limited number of binding sites on an antibody found attached to the surface. Excess free enzyme-labeled antigen is washed off before the substrate is added. The key element in this competitive situation is the amount of color intensity resulting from the substrate addition, wherein the stronger the color intensity, the less the antigen found in the sample. So in the following diagram, we have here the antibody being incubated with the antigen is to be measured, and the antigen is coated with a mixture of antigen antibody. Then enzyme conjugated secondary antibody is added alongside the substrate, which results in a color change, which is then measured afterwards. As we have the direct type of ELISA, we also have the indirect type of ELISA. wherein the ELISA for the antibody detection is very similar to the sandwich antigen technique. Only instead of the antibody being absorbed to the surface, it is the antigen that is being adsorbed. The test sample containing the antibody is then added and allowed to bind to the attached antigen. Unbound antibody is removed by several washings, after which the enzyme-labeled antigen is added. The enzyme-labeled antigen complex binds to a specific type on the bound antibody. When a substrate is added, the enzyme-labeled antigen causes a color change by acting on the substrate. The amount of color change is an indirect measure of specific antibody present in the test sample. So as pictured below, the antigen being tested is coated well, washed, specific body and antibody binds to the antigen, wash again, and then the enzyme-linked antibody binds to the target antibody. Then final washing is done. Substrate is added and converted by enzyme to a colored product wherein the rate of color formation is proportional to the amount of specific antibody present in the sample. To summarize, we have here the diagram of the different ELISA techniques that are mentioned in this presentation, which are the direct, indirect, and sandwich type of ELISA. So, the difference in the mentioned techniques are usually by the process of how the substrate is added to the sample or how the antibodies and antigen are being configured in the sample.
Advantages of enzyme immunoassay. Labels cheap and plentiful. Labels have a long shelf life. Easily adapted to automation. Reaction measured using inexpensive equipment. Very sensitive. No health hazards associated with reagents. Can be used for qualitative or quantitative procedures.
Disadvantages of immunoassay technology. There are initial barriers to getting immunoassay methods accepted for routine use in the environmental community. Under the technical barriers, we have lack of knowledge about analytical options, use of expensive time-consuming methodology when more efficient methodology is available, poor planning of the initial analytical scheme, and failure to identify proper questions to be answered, which can result in generation of data inappropriate to address the problem at hand. On the other side, we have cultural barriers. Under cultural barriers, we have inappropriate or excessive regulatory restrictions on use of new methods, requiring the use of only promulgated methods for program applications that do not have these requirements, requiring the use of expensive broad-scale methods, and GC or MS for limited monitoring applications for only a few known and well-characterized analytes. Enzyme immunoassay has its own disadvantages. Some samples may have natural inhibitors, size of enzyme label limiting factor in designing some assays, non-specific protein binding may occur, and enzyme reactions very sensitive to temperature. This is the last part of our report. Thank you for watching.